the discussion. So this was a okay. This was a the first complete paper that we did. Okay, it had full 50 MCQs, full 4 structured essays, and full 4 essays. Okay, so just from the first 4 lessons, I made it, and okay, I sort of it would have been very thorough. I mean, if you studied for this, and also if you tried all the questions, if it would have been a very good revision and also an active learning exercise of the first 4 lessons. So about 30% of the paper will be composed from the first 4 lessons, right? Then animal form is going to add about another 40%. Okay, just the lesson animal form, and depending on if you are doing the essays, uh, the other lessons will add up to about the remaining 30%. So that's how it goes, no? So yeah, it's important that we keep in touch with these earlier lessons as we move on to upcoming sort of more complicated sections in animal form. For example, we have started circulation already, right? And this week we'll be moving on to things like blood clotting, okay? And then there's blood grouping. We have completed the human circulatory system, I think, in last week. So my point is, it is important that you keep in touch with the previous lessons. Biowala, when a common data, my theory is being fed from the top and it is falling down from the bottom. That is not effective, right? Then all that time you spend sitting on a theory class, listening to a theory class is going to be wasteful, right? If you are bio sort of the theory container has holes at the bottom or the side. Theory is being put on the top and it's falling out. How do you do that? You keep on refeeding it so that whatever is being falling out is again coming inside. So after some time, what we call as systematic repetition, it will stay there. It will stay there. Okay. Just for another 1 year, just for another 1 year, it has to stay there. Then if you can hold it in your brain for 1 year, it will most likely will stay most of it for the rest of your life because it's already in your long-term memory. So if you're doing bio, you're going to go ahead in a certain biological stream, right? Biology associated stream. So it won't go to waste. Anyway, we'll talk slowly. Take the introduction. Sorry, we'll take the start into the paper with the first few questions. Okay, while others are joining. Okay. Okay, keep the chat active, send answers, okay? Right, which of the following polymers are only found in plants? Only found in plants. Glycogen, can you find it in plants? No, it's found in only in animals and fungi. Neither. Glycogen is never found. Then chitin, where can you find chitin? That's in fungal exoskeletons and also arthropod, fungal cell walls and arthropod exoskeleton. Neither. Okay, ribonucleic acid. What do you think? Ribonucleic acid, that means tRNA, rRNA, right? So all types of RNA, they are found in all types of cells, right? Right, because rRNA or ribosomal RNA is the main component of ribosome. So ribosomes are found in all living cells. Ribosomes are found in all living cells. So therefore, rRNA is something that is found in all living cells. Okay. So then inulin, I think that is the answer, right? Inulin is the answer. So inulin is only found in certain plants such as dahlia. So inulin powder is actually used as a sort of a dietary supplement, right, to increase the fiber intake. Okay, because it is indigestible. We can't digest inulin. It adds sort of a bulk. I think we talked about this in nutrition, right? The role of polymers, or sorry, indigestible fibers in your diet, right? It is good for your digestive health. Okay. Keratin, of course, you can find in our body a lot. Skin surface, hair, nails. You can find keratin. All right. So not all. So that's. I think we'll take answer number 4. Okay. Okay. Question number 2. Which of the following is incorrect regarding living cells? Incorrect regarding living cells. Okay, all organisms are composed of cells. That is basic, right? Cell theory. Okay, what are the all organisms are composed of cells? Cell theory, right? There are three components of the cell theory. Basic structural unit of life is the cell. Okay, that's the other part. Basic structural and functional unit of life is the cell. Yes, all cells have a cytoskeleton. Is that correct? Is that correct? All cells have a cytoskeleton. No, usually only animal cells. Oh, out of that, usually multicellular complex animals have a cytoskeleton, right? For example, plant cells don't need one. Okay. So cytoskeleton is usually found in animal cells, okay, as a sort of internal support, but it can be found in certain fungal cells, protozoa such as amoeba, right? Amoeba, right? Sort of certain protozoa, right? Certain amoeba, they have what we call a pseudopodia formation. Pseudopodia formation includes the action of microtubules and actin, okay. So that's false. All organizational levels of matter below the level of cell is not considered as living. Yes. So we are saying it is the basic functional and structural unit. No. So if it is the basic one, then means whatever is below is not living. These are simple things. Okay. Moving on to question number 3, right? May take a basic tikapitagala. We'll quickly go through these things. Okay. Which of the following processes takes place in inner membrane of mitochondria? So, in mitochondria is a elongated organelle. Okay, so inner outer membrane is smooth and the inner membrane is convoluted to form what? Can convoluted to form crystal. Crystal. Crystal of mitochondria. In the living world, whenever you need to increase the surface area, whether it doesn't matter if it's in your digestive system, if it's in your brain, if it's in the inner membrane of mitochondria, where you need to increase the surface area, what do? We have convolutions, right? Rally, right? In your brain, we call them as gyri. Have you seen in brain, there's like wave-like things, right? There's like sort of mountains and valleys, right? On the brain. So that is to increase surface area. In your digestive system, what do you have? You have permanent circular foldings. Circular foldings are there. No? That is to increase surface area together with villi, microvilli. Okay. Okay. Process takes place in the inner membrane of mitochondria. Conversion of pyruvate to acetyl coenzyme. We are talking about the linking reaction here. The linking reaction takes place where? In the matrix. After pyruvates have entered into the mitochondria, then the pyruvate will be converted to coenzyme A by the linking reaction or pyruvate oxidation. Pyruvate oxidation. Production of NADH takes place in the inner membrane. So, okay, Abhi Mulima, what stage of respiration is taking place in the inner membrane? Give me 1 minute. Okay, right. So, what part of cellular respiration takes place across the inner membrane? Yeah, the inner membrane of the mitochondria contains all the machinery, all the cellular components, non-protein needed to convert our electron carriers, right? So that's what our electron carriers, NADH and FADH2, no? Because earlier we used the electrons in glucose and those electrons were given to these to make these. Okay, so they contain high energy electrons, NADH and FADH2. These will be converted back to their NAD plus and electrons, and also H plus, which will combine with uh, oxygen to become water. Okay, so FAD plus and electrons, right? So that is what's happening across this inner membrane, right? Across this inner membrane, this NADH and FADH2, they are getting oxidized, and those electrons are traveling, and that electron energy is actually used to do what? So you don't need this actually. I think I gave you a diagram. They are actually used to pump protons, H plus, into the intermembrane space. They are used to pump H plus, may H plus. They are filling this intermembrane space with H plus. That H plus will come back into the matrix through a special enzyme that we call as ATP synthase. ATP synthase. So when that happens, ATP will be generated from ADP and phosphate. Okay. Um, that's it. No. So actually, think just you can find out what that answer is, and you can just underline that directly, right? Oxidative phosphorylation is what happens. Electron transport chain. You can also call it electron transport chain. Ethanol fermentation doesn't occur in mitochondria because it's part of anaerobic. Mitochondria is only involved in aerobic respiration. Release of carbon dioxide doesn't take place in inner membrane. It takes place in the matrix, right? Release of carbon dioxide takes place inside mitochondria, but takes place in the matrix during the citric acid or the Krebs cycle. No NADH production occurs during the linking reaction, but that is also in the matrix. Understood? Okay. Which of the following? Okay, we'll move on to question number 4. Which of the following stages of the cell cycle does DNA synthesis take place? Hari, then if I quickly remind you about the cell cycle, what do you mean by the cell cycle? Cell cycle is the life cycle of a cell. Cell cycle is the life cycle of a cell. So if you are only thinking about cell division when I say cell cycle, that is not true. Because are you okay? If you are a cell, imagine yourself as a cell. What are, what part of your life are you doing reproduction? Only a small part, right? And if you are a cell, you don't spend your entire life just dividing, right? Cell cycle is the life story of a cell. Okay, so only a part of that life cycle is actually the cell division. So cell division only takes place in the M phase, so the mitotic phase or meiotic venathu, right? That is only about 10% of the cell's life cycle where the cell actually divides. Yeah, the other 90%, okay, the other 90% is dominated by is a, I mean, in the other 90% interface is happening. Interface. Interface has how many subdivisions? Interface, I'm rewinding. Interface has how many subdivisions? There's a G1, S, and a G2. Okay. So interface okay, where? Okay. So we have actually found out the answer. Nothing during the in the middle of the cell cycle, there is no DNA synthesis, no prophase, telophase, metaphase. All those are cell mitotic or mitotic division phases. Of course, the answer is interface. But can anyone tell me which phase of interface DNA synthesis takes place? Yeah, I think very good. Right? S phase is where you make DNA. And is it only what happens in S phase? Right? Is it only what happens in S phase? No. You take the DNA, your double helix DNA, then what do you do? You wrap it around 8 histones. You wrap it around 8 histones to form what? What do you call it when you have DNA wrapped around histones? You form chromatin, right? So chromatin is made during S phase. So DNA is synthesized, histones are made, and then those DNA is wrapped around histone to form chromatin. then that chromatin will stay until it will undergo condensation in the meiotic phase again. First part, what do you call that? Prophase. Prophase again, the chromatin will start to condense. Okay, easy. Okay. Right, let's move on. Question number 5. Which of the following structure function combination is incorrect? Okay, incorrect. Oh, even though in my paper I haven't given correct, incorrect in bold color, in the exam it will be given in bold. Okay, Ardha, it will be given in bold, so don't think about that. So in my papers, be careful because I sometimes forget to bold those. Not sometimes, I always forget to bold those. Anyway, right? Uh, Incorrect. Nucleolus ribosome production. Yes, it produces usually rRNA and other ribosomal proteins and other things. Okay, nucleolus. What do you mean by the nucleolus? There's a dense area inside the nucleus other than the chromatin. Okay, that is involved in these synthesis things. Okay, glyoxysomes. So this is the correct one, right? This is correct. Glyoxysomes, photorespiration in plants. No. So you know what is a glyoxysome? Glyoxysome is a specialized peroxisome. Glyoxysome is a specialized peroxisome. But if it was given as just peroxisome, then this is correct. Yes, photorespiration is done by peroxisomes. Yeah, peroxisomes. I mean, not exactly. Photorespiration is actually this adverse effect of Rubisco reacting with oxygen. Okay, but there's a toxic product that is being produced due to photorespiration that has to be removed, metabolized. So for that, you need mitochondria, chloroplast, and peroxisomes. Okay. So we say, okay, peroxisomes are involved in photorespiration. Glyoxysomes, what do? We say they convert sugar into fatty acids. They convert sugar into fatty acids. You can write on the side, right? Glyoxysomes convert sugars into fatty acids. Yes, it is found in plants, but that is not the function. Photorespiration is not the function. Okay. Cytoskeleton determines the shape of the cell. That is correct, right? Skeleton of the cell. Cyto means cell, right? Anything to do with a cell. Cytology is study of cells. Cytoskeleton is the skeleton of the cell. Cytoplasm is the sort of the jelly-like material inside the cell. Everything is cyto. Okay, if it has to do with cells. Vacuoles store soluble substances needed for cellular activities. Yes. What are the soluble substances? List sugars, pigments, such as what? The example in your syllabus, in your resources, anthocyanin, right? Anthocyanin, those kind of things. Yeah, yeah. Okay. Are stored in your vacuole. Hari, metana, this is not exactly correct, right? Because our product, if you are a sort of a OCD person, this is not exactly correct. Vacuole store soluble substances needed for cellular activities. What vacuole is this more specifically? If you are going for an good A, you should remember types of vacuoles and what vacuole store soluble substances. No, not a food vacuole. Okay, there's two more options. Yes, central vacuole. A central vacuole that is found in plants has this, the structure of a vacuole, right? Vacuole, it's very difficult to draw a vacuole, right? Vacuole. Okay, it is surrounded by tonoplast. Okay, and inside what you have? Cell sac. Okay, central vacuoles store these things. Food vacuoles are mainly involved in what? Food vacuoles are mainly involved in what? Phagocytosis. Phagocytosis. Okay, phagocytosis means the cell is eating. They are the food vacuole
I mean, food vacuole is the sort of the food lunch box එකක් වගේ, right? The food vacuole contains pieces of food and it will combine with a lysosome and digestion will happen. What is the other vacuole that you know in your syllabus? What is the other vacuole that you know? That act as kind of like a kidney. Yeah, for it's like a kidney for these sort of unicellular. Yes, a contractile vacuole. Contractile vacuole maintains the water balance. Okay, so they contractile vacuole sort of remove excess water and solutes. Okay, Golgi complex produces lysosomes. Yes, that is correct. Right, the direct theory parts. Okay, so we can take what is the incorrect one? Answer number 2. Right? Okay, 6. Which of the following is common to lactic acid fermentation, alcoholic fermentation, and aerobic respiration? Okay, which of the following is common to lactic acid fermentation? Okay, alcoholic fermentation. Okay, all three types of respiration. These two are anaerobic. Okay. These two are anaerobic respirations, and there is the aerobic respiration. Common, right? Direct theory points, no? It's glycolysis. Glycolysis. Nothing much, right? Within 5 minutes, you could have come here. Okay, so everything other Krebs cycle happens in aerobic respiration. Electron transport in aerobic. This is linking reaction. This is oxidation of glucose to CO2 and water. What is this? That is like the summary of uh, aerobic respiration, right? You end up with CO2 and water. That is in only in aerobic respiration, no? Okay, so you can't take any of that. Glycolysis is the answer. Okay, right. Which of the following comparisons between C3 and C4 photosynthesis in plants is incorrect? Okay, they're asking the incorrect statement. Let's see. C3, C4. Okay, comparison. So the both statements has to be correct. CO2 fixation occurs only once. That is correct. CO2 fixation occurs only once. Where RUBP plus CO2 catalyzed by Rubisco. Right? Catalyzed by Rubisco. That is the normal start of Calvin cycle. CO2 fixation occurs twice. That is correct. Yeah, the other one is what? PEP plus CO2 catalyzed by PEP carboxylase. Now them I'm just reminding, okay. Again කියද්දි හරි ඔළුවට ඇයි කියලා. Right, what is the product? Oxaloacetate, right? So they're asking incorrect one. Major CO2 acceptor is RUBP, yes. Major CO2 acceptor is PEP. I mean, major. Okay, so it's not incorrect, but you can't say it's not major. I mean, okay, but it's not incorrect. Okay, so major part එක පොඩි issue එකක් තියෙනවා, but we have to go with the best answer. No? So we will see. CO2 fixing enzyme is RUBP carboxylase. Yes, Rubisco. What is the full name of Rubisco? Right? If you take the identity card, what do you have in the Rubisco? RUBP carboxylase oxygenase. No? Rubisco ki full name, right? CO2 fixing enzyme is PEP carboxylase. Yes. So this is the name that Rubisco shows to the world. Okay, I am RUBP carboxylase. I am RUBP carboxylase. But in secret, he is also a oxygenase. Right? The one you don't like to use. So in secret, Rubisco is an oxygenase as well. But he likes to say, okay, I am carboxylase. But in secret, he is doing the oxygenase reaction, which is why photorespiration is happening. Okay, Rubisco. Then photosynthetic yield is high. No, Usually C3 plants require very specific conditions unless provided they can be inefficient because of what? Can they can be inefficient because of photo respiration. Photosynthetic yield is usually low, that is false. Okay, usually C4 plants work in a wide range of conditions, wide range of temperatures, wide range of CO2 conditions, wide range of light intensities. C4 plants can work. Okay, they are more versatile. Okay, okay. Then finally, first product of photosynthesis is PGA. Uh, First product of photosynthesis. So we can say, I mean, okay, so it's not the first product. You get the 6 carbon unstable, which will break down into two molecules of 3 PGA. 3 PGA will convert to G3P. Right? I'll remind you. Okay. What is the Kelvin cycle process? I think if I can show you the new 2028 tute, I can revise you from that directly. Our 2028 people, the tute I have given them, I have included. I think for you, the tute was slightly different. I already had it. Yeah. Okay. So let's see. Okay. What are we talking about? Photosynthesis, no? Kelvin cycle. Okay. So did I give you this diagram in when we were doing tute? Okay. So quick revision of Kelvin cycle, Taggalahaganna, right? Input is in the form of carbon dioxide, but for the sake of the cycle, we are considering three carbon dioxide at the same time. So this is your CO2 acceptor, no? RUBP is here. RVP has 5 carbons, 1, 2, 3, 4, 5. And for that, we are going to add the carbon of carbon dioxide, and you are going to get a 6 carbon short lived intermediate. Okay. Then that will break down into 2, 3 PG. Okay. Then you have G3P, 1,3-bisphosphoglycerate. G3P. G3P, part of the G3P will be sent to part of the G3P. Sorry, so this is there's an intermediate as 1,3-bisphosphoglycerate, and from that 1,3-bisphosphoglycerate, you don't need to know that. Okay, there we make G3P. Part of the G3P will be sent to make sugars that will actually become the glucose now. Then another part of the G3P will be used for the regeneration of RUBP again, because the cycle has to continue again and again and again. Okay, so I hope you can remember that. I always find it helpful when you remember a certain diagram whenever you are trying to uh, recall, right? Whenever you are trying to recall something, take the help of a diagram. The first product of photosynthesis is oxaloacetate. Yes, I already showed you here, right? PEP is going to combine with carbon dioxide to produce oxaloacetate. Okay, all right. The levels immediately, question number 8. Okay, question number 8. Levels immediately above and immediately below the basic structural unit of life in the hierarchical order of biological organization in correct order. Okay. Basic structural unit of life means the cell, right? Right? They have saying, okay, immediately above and below the level of cell. So you have to find out that also, right? Above and below the cell, you have to find out. Okay, simple question. There's only 4 answers. Yeah, there's only 4 answers. Anyway, right, that makes it much easier. No? What is above the level of the cell? Yeah, what is above means what do cells get together to form? Tissues. Cells have their in their inside they have organelles. Answer number one is answer number one. Immediately above you have so you can take above and below interchange. Hold on. Immediately above is tissues. Immediately below is organelles. Yeah. So that's a small issue. If you have marked anyone who has marked answer number 3, anyone who marked answer number 3, hands up or send me a response. Yeah, yeah. You can take a mark for that. Okay, but doesn't mean answer number 1 is also false, right? You can take either answer 1 or 3 both. Okay. Yeah. Okay, because it's given asking in the correct order also. No? So if you take as going from the lowest level upwards, you have to start from the simplest level from your organic molecules, and then you make organelles, cell, hemanian. Okay, that's fine. Take 1 mark. Okay. All right. Some major polysaccharides and their building unit are shown below. Question number 9. Okay. Any questions? Okay, so they're asking the correct one. So the question is not question number 9. Okay. All right. So question number 9 and 10. There's a question missing, no? Yeah. Nobody mentioned this that there's a question missing. Yeah. Okay, during the exam also. Okay. Anyway, some major polysaccharides and their building unit. This is not question number 10. Okay, it's still question number 9. So 10 is missing. Okay. So for that, I think they have given. I think we gave marks. Yeah. Question number 10 is missing. We have given the bonus mark for the missing question number 10 also. Okay. Correct recipe represents the polysaccharide and the building unit. Okay. Right. Let's quickly match. Inulin is fructose AR. Pectin is composed of what? Pectin. Yeah. Yes. There's an answer there, right? Question number 9 or the 3. Okay, yeah. Pectin is composed of galacturonic acid, okay. Chitin by glucosamine, okay. So then cellulose is the one remaining, DQ, okay. Answer number 3, no, A, R, B, P, C, Q, D, S, okay. Simple enough, right? We'll quickly go through these things, okay. Any questions? All right, question number 11. Golgi apparatus, a stack of network of flattened and tubular sacs. What are we describing here? A stack of flattened and tubular sacs. We are actually describing here endoplasmic reticulum. Right? Because if you take your RER, it is flattened, and tubular is SCR. Your Golgi is like a flattened stack. Okay. In question 8. Yeah. So it should be answer number 3. That clarified you, right? Okay. Transphase receives vesicles from ER. No, right? If you draw, I think it's easier for me to just quickly show you, right? It's easier for me to quickly show you. This one. Yeah. Okay. What are the two phases of Golgi? What are the two phases of Golgi? Methi? Okay, so you have the nucleus in the center, then it immediately surrounded by rough endoplasmic reticulum. Then from rough endoplasmic reticulum, you have the tubular smooth endoplasmic reticulum arising. Then rough endoplasmic reticulum and smooth endoplasmic reticulum, they produce vesicles, no? They produce your phospholipids, and also RER is the main protein producer. Okay, and then you make fats, lipids, oils, blah blah. Everything will be packed into vesicles. So you have a Golgi very nearby. Several Golgi can be there actually. Golgi can be there. Particularly Golgi amount is high in secretory cells. Right? So this is the cis phase. Can you see cis Golgi? This is the cis phase. Cis phase, cis receive. Cis is for receiving. Cis phase is the receiving phase. So the content. So if it's a Golgi is a post office, it is a where you accept parcels baragarna counter have you seen, right? Then it will travel through the Golgi and they will be repacked, reorganized, and it will be sent away through the trans phase. There's a trans, the other side is called the trans Golgi. Okay, clear? Trans phase doesn't receive, you can cut and correct. Cis phase is known as membrane factory. Who is known as membrane factory? You can write the correct answer, right? Who is the membrane factory? RER, rough endoplasmic reticulum. It synthesizes phospholipids. Okay, is abundant in secretory cells. Very correct. Yes. Autolysis causing cell death. Who is causing cell death? Suicide. Cell suicide. Who helps the cell to commit suicide? Yes, lysosomes. Lysosomes are very dangerous, right? It's like a person owning a gun can die from its own gun, right? He can accidentally shoot himself, right? Lysosomes are found inside the cell to help in digestion, right? I'll show you. That is what we call as phagocytosis, no? Right? That is what we call as phagocytosis. So during phagocytosis, what's going to happen? Did I have endoplasmic reticulum? Okay. Yeah, maybe. Phagocytosis, what's happening? There's a piece of food that is near the cell, and that piece of food will be engulfed into the cell, forming what we call as a food vacuole. Okay. Then there's a lysosome coming, and that lysosome contains digestive enzymes, as you know, and they will combine with the food vacuole, and the digestion will happen, and whatever things needed by the cell will be absorbed, right? Then I have given, autophagy. Autophagy, the word is not there in your resource, but autophagy is the recycling of old organelles, right? For example, then not in Sri Lanka, in other countries, when a vehicle gets old, we send it to be recycled, scrap kernel. That's like that. So, for example, there's a mitochondria that is old, not functioning, or it's like damaged, not very effective. So what we are going to do is you are going to combine it with a lysosome, and that lysosome is going to break down that whatever old organelle, and it will those sort of the raw materials will be used to make new organelles. Okay, so that's that. Okay, what's the answer? Sorry, what's the answer? Answer number 4, right? Abundant in secretory cells. Right. Question. Moving on to question number 12. Okay. Any questions? Some of the stages and events of meiosis in a plant cell. Okay. Specific. Let's see. Stages and events. Prophase 1. Nuclear envelope breaks. What do you think? In prophase 1, nuclear envelope breaks. Yes, right? Nuclear envelope fragments and the nucleolus disappears. All those things happen in prophase 1. Okay. Prophase 2, centrosomes start producing the spindle apparatus. Centrosomes start producing spindle apparatus. What do you think? What's the problem? There's no spindle, right? There's no spindle in plant cells, right? There's a microtubule complex, accumulated microtubule complex. Am I correct? Right? That is going to be the chromosome mover, right? So in plant cells, the spindle is not very properly organized, right? In animal cell, that is not properly composed of the asters and the centrosomes. Okay, so spindle is not found in plant cells. That's why this word is important. Metaphase 2, genetically identical though chromosomes are arranged on metaphase plate. No need. What are we talking about here? Meiosis. In meiosis, because of what? Crossing over, what do you end up with? Genetically non-identical. No? Crossing over, you end up with genetically non-identical. Then anaphase 1, sister chromatids move towards opposite poles. Anaphase 1, what happens in anaphase 1? So anaphase 1 is a part of meiosis 1. In meiosis 1, do you separate sisters? No. In meiosis 1, you only separate homologs. Okay, only homologous pairs are separated. So when that happens, sisters stay together. Sisters stay together. Therefore, you can't say sister chromatids move towards opposite poles. Okay, so I'll show you with a quick summary of. Yeah, in meiosis, what happens in anaphase 1? You can see the homologous pairs have been separated. So the blues and reds have been separated in anaphase 1. Got it? And the sisters will be separated in anaphase 2. Sisters are separated in anaphase 2. Sister chromatids. Right? But also the sister chromatids are no longer actually kind of not like sisters, because why? Because of the crossing over, they are unidentical, because of the crossing over. You can see blue chromosome is having pieces of red, and red chromosomes are having pieces of blue. That is because of crossing over. So they are unidentical, genetically non-identical. So the answer is actually only A, ne? A only. Any questions? Okay, shall we move on? Any doubts? Okay. Right, let's move on to question number 13. The monomer structure of a structural linear polysaccharide. The monomer of a structural linear polysaccharide. Right? So if you take fructose is a storage, no? So then ethylene eka, that is out. Right? Sorry, hold on. Fructose forming inulin. Okay, right. The monomer, yeah. So what I meant to say is yes, fructose forms inulin, no? Right? Inulin is storage, so that is out. Pentoses and hexoses, what are we talking about? Hemicellulose. Right? So hemicellulose is structural, but that is correct. Hemicellulose is linear, right? No, wait. Hemicellulose, we don't take, it could be branched or linear, right? Hemicellulose. Okay. Is there a better answer? Structural linear polysaccharide. Okay. You can take amylose. Amylose is composed of glucose, no? Right? So that is the best answer, right? Any other answers that you had uh, considered? Any other answer? Galacturonic acid, pectin. Yeah. Any other answer that you have considered? Yes. So best answer is amylose. Amylose is a linear structural polysaccharide that is composed of glucose. Yes. Hemicellulose is given as branch. Yes, I had to double check. Yes, hemicellulose is a branch. No, it's branched, so you can't take that. Okay. Clear? Question number 14. Medical Taggala Virakala will go into deeper details about plant form certain questions. Okay. Correct statement regarding plasma membrane. Mainly composed of carbohydrates, phospholipids, and proteins. No, no, these are basic knowledge, right? It is a mainly phospholipid bilayer with some proteins embedded. Neither, so there's nothing. I mean, it has carbohydrates, but not majority, right? Not majority. Transmembrane proteins act as pores that allow ions and some non-polar molecules to pass through. What do you think? Some non-polar transmembrane proteins act as pores that allow ions and some polar molecules. Ne? It allows some polar molecules. So that is force, right? Then the composition and function of two sides of the membrane are the same. No, there's an extracellular side and an intercellular side. They are modified to function in the two environments, right? Extracellular side has interaction with other cells, other molecules. Okay, it could be just the outside. If you are a if you are a cell on the external surface, you are a cell that is interacting with the outside. So the outer environment is completely different to the inner environment, right? So therefore, their composition is different. Since it is selectively permeable, it regulates exchange of substances. Yes, plasma membrane is a very good filter, right? It's a very good filter because of the selectivity of certain transport proteins. Only things that are needed for the cell are supposed to enter. There are things such as viruses that can overcome these barriers, okay, and certain bacteria by producing enzymes and toxins that can break down the plasma membrane, they can enter, but usually for substances, not organisms, for certain molecules, certain ions, if it's, I mean, if you are not allowed to, or if you don't have a transporter, then they can't enter. Okay. Cholesterol molecules provide the fluid nature. Yes, there are some cholesterols here embedded here and there. Can anyone tell me in what kind of cells? Cholesterol is only found in animal cells. Okay, that's a MCQ point you need to remember. Plant cells, how do they maintain their membrane fluidity? Plant cells, how do they maintain membrane fluidity? Yeah. Plant cells, how do they maintain? We talked about that when we are talking about plant response to cold stress, right? So when the environmental conditions become cold during freezing, there's a threat of freezing. What can a cell do to maintain its plasma membrane in a normal fluid state? Fluid means sort of a permeable normal state. What can it do? This how the plant and animal cells maintain their plasma membrane fluidity. Can you see? Cholesterol is found within the animal cells. Cholesterol is found within the animal cells. So you can see the cholesterols that are here and there. They prevent the sort of the they prevent the sort of the packaging, tight packaging of the phospholipids. If you come to plants, they have actually, I mean, what they do is they increase the proportion of unsaturated. Yes, they alter the lipid composition. They increase the proportion of unsaturated fats, unsaturated fatty acids. So what happens when you have unsaturated fatty acids? There's a double bond somewhere. No, whenever you have a double bond, they bend. So can you see? Because of that bending, it is sort of impossible for the phospholipids to pack tightly. Okay, so that is going to prevent freezing. Okay, clear? Visually remember. In animal cells, you put cholesterol in between, but in plant cells, you increase the proportion of unsaturated fatty acids found in the phospholipid tail. Okay. Okay, moving on. Come back to the paper. Okay, 14, that is done, no? Answer number 4, right? Okay, 15. Let's see. Correctly measures subcellular component and function. Okay. Golgi produce peroxisomes? No. Golgi doesn't produce peroxisomes. You can say it produces lysosomes and also uh, sort of the cell plate vesicles, the vesicles that form the cell plate, right? Those are produced by Golgi. Lysosomes digest worn out organelles. What do you call this? We call this autophagy, right? You don't need that. Okay, that is correct. Smooth endoplasmic reticulum synthesize membrane phospholipid. You can write down the correct answer. That is rough endoplasmic reticulum. That does this. Okay. Rough endoplasmic reticulum in detoxification? No. Usually detoxification is by smooth endoplasmic reticulum. Okay. SCR. Cytoskeleton can move fluid over the surface of tissue. Who are we talking about here? Can anyone tell me? Moves fluid over the surface of tissue. Someone we'll encounter this in when we are doing the respiratory system in your respiratory epithelium. Oh no, we talked about tissues. No, what is the respiratory epithelium? Pseudostratified columnar ciliated, no? Ciliated pseudostratified column. All right. So that is the epithelium that you can find in your upper respiratory tract, in your trachea, and also the branching tubes leading into the lungs. Right. So whenever you have some things, dust particles that are in the mucus, right? There's a sticky substance that is covering your respiratory tubes. Okay. So you are breathing in a lot of dirt, right? Dust, viruses, bacteria, fungal spores. So many things you are breathing in, all those things will be trapped in the mucus. But if that mucus, if you are trapping things in mucus, you have to clean them, right? If you just trap them in your respiratory system, that is even worse. You are holding them in one place, very good chance for them to cause damage. You have to somehow remove them. Atugala අයින් කරන්න ඕනේ. That is done by cilia that is beating in a nice rhythmic manner. Okay, clear? So 15, answer number 2. 16, which is following is correct about the central vacuole found in a plant cell. Okay, so correct about the central vacuole. See, they have given the proper full name. We are talking about a central vacuole, not just another, I mean, not the other two types of vacuole. Stores water and other materials such as ions, sugars, and pigments. That is exactly correct. Okay. The composition of the cell sap and the cytosol is similar. No. Cytosol contains what? Cytosol can contain so many amino acids, sugars, all the other sort of the cell sap, and the cell sap mainly contains these, right? Sugars, ions, and pigments. It is more watery. Cytosol is more jelly-like, right? It contains so many enzymes, right? Small other molecules. It's packed full of so many things, right? Cell sap composition is much more simple. That is the fluid found inside the vacuole, cell sap. Okay. Sac pigments give turgidity and support. No. What is the pigments are color? They give the color. What gives the turgidity and support? It is the water in the vacuole, right? The water of the cell sap. So if you take like a plant cell, when we talked about this when we are doing water potential, right? What do you mean by the turgidity? What do you mean by turgidity? Because if you take a plant cell, it is kind of dominated by the central vacuole. It is kind of dominated. So all the other sort of organelles and this nucleus and mitochondria, all the other things are sort of found on the side. So when the vacuole gets filled with water, that water exerts, sort of expands the entire cell. Okay, so therefore that exerts a push on the cell wall and the cell wall pushes back and you have a turgidity. Water insoluble pigments, that is no, right? Water soluble pigments are found. Anthocyanin is correct, but it's a water soluble pigment. Right? You can correct it. Ions such as calcium, you don't store calcium much, you store sodium, right? Sodium and chloride. Okay. Okay, what is the test given here? Identify proteins. Shall we move? Question number 17. What is the name of the test that we are doing here? Identify proteins. One other color. Okay, aqueous protein solution you have taken. Copper sulfate dala, KOH dala. What is the correct order? Yeah. This is the biuret test. Yeah, the biuret test. Did I show you the biuret test in earlier in the practicals that we did? Did I show you biuret test? Yeah, I think so. Yeah. And if you go to my YouTube, there's a much more better video of the practical seminar I did for the 28 people. It might be a bit too long. You can in the right in a lot of practicals ටික revise කරගන්න small, I mean small video, only about 3 hours. Okay, right. No need to watch that. Okay, just read the practical guide. Okay, you don't need that much elaboration. Taggala practical guide, that's fine. Okay, so you add You take protein, you add KOH, then you add copper sulfate. Okay. Clear? This is a bioret test. You add KOH and then you add copper sulfate. 5% KOH, yes, and 1% copper sulfate. That is exactly given. Okay. Any questions? Is it K plus that is found in? Okay, that is okay. K plus. Oh, okay, yeah, yeah. In the cell sap, okay, yeah. In cell sap, you can find K plus, right, of the central vacuole. Okay, yes, right, yeah. This found it's not sodium. Okay, sorry. That's good. Okay, you can find ions such as potassium and Cl minus. Okay, that's good. Right. Question number 18. Correct statement regarding photosystems. Photosystems. Question number 18. Light harvesting complex contains only chlorophyll A and chlorophyll B. No, you have other accessory pigments, ne? And xanthophyls, right? Carotenes, all those are found. So that is false. You can add carotene, xanthophyll, those are also found, right? Light harvesting complex. Okay. Primary electron acceptor in photosystem 2 involves in both linear and cyclic electron flow. No. Photosystem 2 is only involved in linear. Who is involved in cyclic electron flow? That is your photosystem 1. Right? That is your photosystem 1 that is involved in both. Photosystem 2 is not involved. Chlorophyll A in photosystem 2 is known as P700. No, you can correct it. Who is he? P680. A special chlorophyll A molecule in photosystems absorb photons and become reduced. In photosystems, absorb photons and become reduced. What do you think? No, right? Chlorophylls, they oxidized. No? So if you can draw roughly a reaction center, there's two chlorophyll molecules. What happens when you give them when you give them the energy of photons? What happens? They release electrons. They release electrons. So that is an oxidation, right? They absorb the energy of photons. They become oxidized, and that those electrons will be accepted by the primary electron acceptor. That's what happens. Electrons released during hydrolysis of water is used to neutralize the excited photosystem too. Yes, right? This is where the splitting of water happens. Okay, so H2O will split to produce H plus O2 and electrons. These electrons will be used to neutralize the chlorophyll A. Therefore, oxygen gas is produced during photosynthesis. So the oxygen that we so desperately need, okay, that is a byproduct of photosynthesis. Just what is the plant? What does the plant want? The plant only needs the electrons. And as a byproduct, it produces oxygen, and that oxygen is precious to us. Okay. Any questions? Okay, right. Let's continue. Can you hear me? Yeah. Right. Common compound to both photosynthesis and cellular respiration is common compound to both cellular respiration and photosynthesis. What is it? You can directly answer. Ned. What is produced in both photosynthesis and cellular respiration? Yeah. Pyruvate, no? Pyruvate. Okay. FADH2 only cellular respiration, NADPH only photosynthesis, 3PGA only Calvin cycle, acetaldehyde, that is it lactic acid? Yes. Okay. Next question. Okay. Question number 20. Which of the following is correct regarding the event related to the evolution of biological diversity and corresponding time period? Okay, we are moving on to a bit of evolution. Okay, so question number 20. 3.5 billion years ago, origin of the first photosynthetic organism? No, right? That is actually the origin of the first prokaryote, right? These are direct, nothing to be explained, right? Evolution of sponges, I think that is the answer, right? 700 million years ago, sponges evolved. Okay. All the others, direct, no, shall we, no need to fixate on that. You can find out the answer. So the answer is number 2. Okay. 21. Correct statement regarding evolution of seedless vascular plants. Okay. Correct statement regarding evolution of seedless vascular plants. Question number 21. According to fossil records, ancestors of vascular plants had gametophytes and sporophytes that were equal in size. Yes, so that is how evolution happens. No, so in our modern environment, we have usually gametophytes that are reduced and sporophytes that are usually dominating, right? So, but earlier, as evolution was happening, gametophytes and sporophytes were equal in size. Because when we move on to vascular plants, I mean, in non-vascular, of course, in your bryophytes, you have gametophytes that are larger in size. But when you jump to vascular plants, immediately you start to have compared to the sporophyte, the gametophyte is reduced. Okay. Producing a male and female gamete by the same plant is called heterospory. No, right? By the same male and female gametes by the same plant, that is, you can call as bisexual, right? Heterospory means you would produce 2 spores. Those 2 spores give rise to a male and female gametophyte. Spores give rise to gametophytes, right? So that is not what we mean by heterospory. False. Xylem vessel elements in the plants transport water and minerals efficiently. Xylem vessel elements. Do you have xylem vessel elements in seedless vascular plants? Can anyone tell me? Do you have xylem vessel elements in seedless vascular plants? Yes? No, right? They are only found in, yes, seed plants. They are only found in seed plants. Xylem vessel elements are only found in seed plants. All the others, they have to just survive with tracheids. They only have tracheids for their water transport. Vessel elements are only found in, you can't even say all seed plants, because only some gymnosperms have vessel elements, right? But you can definitely say they are found in all angiosperms. Okay. What is the gymnosperm phylum with xylem vessel elements? What is the gymnosperm phylum with xylem vessel elements? Yes, phylum Netophyta. Right, phylum Netophyta is the only gymnosperm phylum that has xylem vessel elements. All the other gymnosperms do not have. Gymnosperms can walk the naked seed. Then angiosperms all have, but xylem vessel elements are not found in seedless vascular plants. All club mosses are heterosporous, and the spike mosses are homosporous. පැත්තමාරු කරලා දීලා තියෙන්නේ නේද? Okay. They have given it the other way around. All club mosses are homosporous and the spike mosses are heterosporous. If you just turn it crisscross කරාන, all spike mosses are heterosporous, club mosses are homosporous. Similar tissue arrangements are found in stems and roots of early vascular plants preserved in fossil. Early vascular plants, stem and okay. Do you have any mention of that? Stems and roots of early vascular plants preserved in fossil. Do you have any mention of such thing? No, right? Yeah. So the answer we can take as answer number 1. Okay. Question number 21. Answer number 1. 22. Cell wall components, photosynthetic pigments of some organisms of kingdom Protista are given below. Okay. We have to select the correct combination. So Gelidium, diatom, sargassum. Okay. Then you have the photosynthetic pigments of each of them. We have to match. Okay. So let's see. Gelidium. Gelidium, what is the, they are called as red algae. Ne? Gelidiums are called as red algae. Okay. So what is the storage, cell wall component? Gelidium, it is mainly cellulose, no? So you can match gelidium and cellulose. So AQ, there's a lot of answers. So we'll go ahead. And what is the storage sugar of gelidium? It is, photosynthetic pigments. Right? So what is the photosynthetic pigment? You have chlorophyll A and phycobilin. So Z, no? Z. So AQZ, there are two answers. So we have to go ahead. B diatoms, diatom cell wall component. Pectin, no? Pectin. So they have given the not the obvious one, the one you can remember. Okay, anyone can remember. Okay, diatoms have silicon. They have given the hidden one, right? Pectin diatoms contain pectin. What is the photosynthetic pigment? Carotene. Sorry, chlorophyll A, C, carotene, xanthophyll. Yeah? So BRX. Okay, so we have found out the answer. Answer number 2. Yeah? C, sargassum, alginic acid. Okay, because that's how you extract. Usually we use gellidium to extract agar, right? Agar, we'll talk about agar uses when we are doing microbiology. We use it to prepare cultures, microbiological cultures we make using agar. Okay, it is extracted from gellidium. Any questions? Any questions? 23. Which of the following combinations is correct regarding organisms in phylum Nidaria? Okay. They do have gastrovascular cavity. They do not have protonephridia. No excretory, circulatory, nothing. Okay. Nematocyst. Yes, very correct. Nematocyst is the stinging capsule, right? They contain the stinging thread, right? And mesoglia. What do you mean by mesoglia? Is instead of the mesoderm, then if you draw a very simple nidarian, so this is the, so these are the tentacles, and what are the two layers? So you can say this blue color layer is the ectoderm, right? Blue color layer is the ectoderm, and there's an inner layer of what we call as the endoderm, and in between the ectoderm and endoderm, it's supposed to be the mesoderm, but because these are what, diploblastic, nidarians are diploblastic, they don't have a mesoderm, they have a mesoglia. So that is correct. Sensory papillae, no, those kind of things are not found. Sensory papillae, can anyone tell me sensory papillae are found in annelids, right? Diploblastic, that is correct. Papillae, annelids, mesoglia are correct. Mantle, something found in, yeah. Mantle is something found in mollusks. Okay. Tentacles you can find in Idaria, but both. Okay, let's move on. 24. Which of the following combinations is correct regarding characteristics of organisms in phylum Chordata? Okay, chordates. 24. Carp. Carp. What type of a fish is carp? Yes, what type of fish is carp? Carp is a bony fish, Osteichthyes. Right? So, what do they have? Can anyone remember what type of scales do bony fish have? Bony fish? Bony fish, everything is sort of bony, right? So their scales are close to bony structure as well. So, Osteichthyes, what kind of scales should they have? If you can remember, then when we are talking about our mammalian bone, we talked about how bones are composed of concentric circles. Ne? Bones are composed of concentric circles. So, that is exactly the same arrangement that you have in the scales of bony fishes. We call them as cycloid. Cycloid, or there's a ctenoid scales, it's a different type, right? So, cycloid scales are actually having the sort of the arrangement of bony, sort of bone. So, placoid scales are very close to teeth. Placoid scales are මෙහෙම නිකන් sort of teeth වගේ. That's the scales that you can find in sharks. So no, you can correct it. They have placoid or cycloid scales. Okay. Then bat cloaca. Cloaca is something that is found where? Cloaca is something that is very interesting in your sharks, right? Sharks, or then I mean you can say chondrichthyes, cartilage fish. In them, they have a very, I mean, a common opening for all three things: for the genital opening, for the excretory or the urinary system, and also for the what digestive tract. All open into a common opening. Then there it will open to the outside. Cloaca, you can write in front. It is found in chondrichthyes. Lizard is a reptile. Lizard is a reptile. Does a reptile need a skeleton with air cavities? No. Why do you have air cavities? To reduce the weight of the skeleton. To reduce the weight of the skeleton. So that is something we needed for flight. Okay. So that is aves, birds. Eagle color vision. What do you think? Yeah, sort of birds have very good eyesight. Okay. Birds have eyesight with color vision. So eagle, that is the correct answer. Cow, non-glandular skin? No. Cows are mammals. Mammals have skin with glands. Okay. Clear? Going too fast. Let me send me a response or sleeping, whatever, what's happening. Right? Going too fast. No. Okay. I have the 26th paper class at 8. Okay. So we'll see the maximum we can cover before 8. Okay. Right. So these days we are in Apeksha Hospital. So kind of busy. But it's interesting to see how many people are living with their cancers. It's kind of normal to them, right? The entire hospital is dedicated to cancer, and so with time they started to adapt to their problem, right? Start living with their issues. So that's why, I mean, it's kind of busy because there's a lot to learn. Okay, and I mean it's not just cancer. Cancer will current radiotherapy, chemotherapy. That's not just it. Right, there's a lot of things. There's a lot of surgeries involved. There's a lot of medications involved, and cancer patients have all the other diseases, so they have to manage that as well, right? So people in the cancer field are very sort of they are, let's say, they are experts in so many fields. If you want to treat cancer, you have to know so many things about other fields, right? So it's a interesting appointment that we are doing these days. Okay, anyway, shall we move on to 25? Apoplastic root. Okay, question number 25. Any questions up till now? Apoplastic root. So apoplast occurs through the continuum of cell walls and intracellular spaces that includes the lumen of dead cells also. So always think of apoplastic as going through dead things. So cell wall is not a living structure, no, right? So is there something facilitated diffusion happening? In order for facilitated diffusion to happen, you need to have special what? You need to have this sort of transmembrane proteins, right? Okay, so that is something found in the plasma membrane. In the apoplastic route, plasma membrane is not involved. But bulk flow, what do you think about bulk flow? Is something bulk flow, is apoplastic route, I mean, does bulk flow happen by apoplast? What do you think? Yes, in the xylem ascent or xylem transport, xylem vessel elements are dead cells, and inside that lumen is part of the apoplastic route. Yeah, that lumen is part of the apoplastic route. So bulk flow is part of apoplast. Yes. Osmosis and diffusion. What do you think? Transport of water along apoplastic route. Is there a diffusion taking place? Because they are asking water. Read the question very carefully. Do we talk about a diffusion for water? Do we talk about a diffusion for water? No. We give the diffusion of water a special name, that is osmosis, right? So in a situation where, okay, in a situation where there's two liquids, okay, let's say two water soluble, I mean water based liquids are there, okay. On one side there's a lot of molecules or solutes. On one side there's a lot of solutes, and it is separated by a membrane that is impermeable to that solute. And on this side, there's only a small amount of solutes. You know, imagine a situation where two solutions are separated by a membrane that is impermeable to the solute, my red color solute. So, what should happen? The concentrations should equalize. No? So, in order for the concentrations to equalize, what is the only option? The fluid has to go to this side and increase the volume of this side so that it can become more dilute. So the movement of the solvent, okay, movement of the solvent is called osmosis. Osmosis is the word that we use for the movement of water along a concentration gradient. Okay. All right. So diffusion doesn't take place. Osmosis and bulk flow is very correct. Okay, bulk flow is the thing that we include in the xylem ascent and xylem transport. Facilitated diffusion doesn't take place. Okay, any questions? Did you do question number 26? Okay, that's a bit of a water potential experiment. Our petiole story. Here is it. Alocasia petiole strips with 160-degree angle of curvature was immersed in two sucrose solutions, X and Y, kept in equilibrium. Observations were taken after 1 hour. Okay. The curvature of the petiole strip in solution X was increased while the curvature of the strip immersed in solution Y was decreased. Right. What is the theory behind this? What is the theory? So once you cut the alocasia leaf petiole, it is going to open up. with the sort of the inner fleshy part facing outwards. Ne? It is going to open up like this. No? Katai. Okay. It is going to open up so that the epidermis is outside. So this is the blue color is the epidermis. And the green color, this is the sort of the fleshy or the parenchyma. That's going to open up. Then if you can mark 3 points, method point, there's a point here and there's a point here. And if you draw lines like this, and you measure this angle, you can take it as the angle of curvature. Okay. Then what are we doing? If water comes in by osmosis, if water comes in, what's going to happen to this angle of curvature? If water comes in, what's going to happen to this angle of curvature? Because of the volume increase of the parenchyma cells inside, what's going to happen? This angle of curvature, can anyone tell me what is going to happen to the angle of curvature? What's going to happen to the angle of curvature? It creases. What creases? Angle of curvature. Okay. When water comes in, parenchyma cells increase in size or swell. What's going to happen to the angle of curvature? It decreases, right? This curvature, you can obviously see this is less than this. Decreases. Yes, very good. Then the other option, what can happen is, if water goes out, if you lose water from this tissue by exoosmosis, the cells are going to shrink, and this tissue is going to become more straight, right? The tissue is going to become more straight. So the curvature, what? The curvature decreases. This is the theory part behind this, no? The curvature decreases. If water comes in, the curvature decreases, and curvature decreases. Therefore, the angle. The curve gatiya වැඩි වෙනවා, curvature increases, but the angle decreases. Here, if you lose water, the leaf petiole becomes more straight. So the curvature decreases, but the angle increases, right? The angle increases. There. If I repeat, if water comes in by endosmosis, cells are going to become bigger so that it is going to curve more. Curvature increases, but as the curvature increases, the angle gets less. Water goes out, the swells become less, the cells shrink. So the tissue becomes more straight so that the curvature decreases, but the angle increases. Okay? The curvature of the strip immersed in solution X. Okay? Curvature of the strip immersed in solution X increased, solution Y decreased. Then now you can assign what happened. Curvature increased. Curvature increased in solution X. So in solution X, when you put it in solution X, what has happened? Water has come in. The curvature increased. Be very careful. Kiyanne angle of curvature, the just curvature, okay? Then the curvature of the strip immersed in the solution Y was decreased. Curvature decreased because it became more straight. That is because of losing water. The tissue lost water. Exoosmosis. Okay? So what is correct? Then tonicity. Matakaid, right? What do you mean by a hypertonic solution? What do you mean by a hypertonic solution? Means tonic means solutes, right? Hypertonic means it contains a lot of solutes. When you have a lot of solutes, its solute potential is very high, but solute potential is a negative value. Therefore, water potential is a very low value. So hypertonic solutions are dehydrating solutions. That means if you put something into hypertonic, it is going to suck out all the moisture, suck out all the water from a tissue. If you put something in a hypertonic solution, because why? It has a lot of solutes. Its water potential is less. Therefore, water is going to come out of the tissue into the solution. This is hyper. What is about hypo? Hypo means it has less solutes, therefore it has more water, therefore its water potential is much high. If you put something in a high water potential solution, the water is going to come inside the tissue. Okay, so you can say Y and X. So we identified. So what has happened? In X, water has come in. So you can say X is a hypotonic. Y in Y, you have lost water. You can say it's a hypertonic solution Y. Can you understand how we arrived at that? So let's see. Solution X is hypertonic compared to the tissue. No, you can correct it. It is hypotonic. Endoosmosis has taken place in the tissue immersed in solution Y. No, in solution Y, what happened? You lost water, so it is exosmosis taken place in the solution Y. That's why the tissue became more straight. Water has moved out of the tissue immersed in solution X? No. Water has moved in by endoosmosis. Water potential of the tissue is higher than the water potential of solution Y. What do you think? Yes, because we said Y is a hypertonic solution. So if you are losing water, that means water is moving from high water potential to low water potential. So the solution has low water potential, tissue has high water potential. That's why the tissue is losing water. Answer number 5. Water potential of the solution X is lower than the water potential of the solution Y. No, because we can clearly compare them. No, you can clearly say this is a hypertonic solution. Hypertonic solution is Y. Okay, Y is a hypertonic solution and X is a hypo. So that is false. Good question. Let me know if you can't understand this. It's a good question. Let me know if you can't understand question number 26. Do you want me to explain again? Anyone? Raise your hand. Okay, moving on. Sleepy, no, right? Question number 27. So, water potential concepts, I mean, because as I keep on telling, you are facing the exam bit closer to the end of the life of a syllabus, right? Syllabus again, antima kala. So they think after 2030, the syllabus there will be a syllabus change. The syllabus was introduced in 2017, or if you take realistically, right, 2018 it was properly established. Okay, so almost 10 years. So they have run out of sort of certain question templates or question patterns to be that can be asked from this syllabus. So they can ask certain novel things from practicals, from practical concepts, right? So that weight can increase in your 27, 28, 29 years. Okay. So that's why I mean, I include these questions. Sometimes they can be a bit excess, but it's good to practice them now when you have time rather than close to the exam. How is it? 27. Okay. Uh, Incident that occurs in metaphase of mitosis. Nuclear envelope fragments and chromosomes get even more condensed. Which phase does that happen? Which phase the nuclear envelope ruptures in mitosis? Yes, can anyone tell me? In which phase of mitosis does the nuclear envelope fragment? Very good. Prometa. This is talking about prometa, not meta. Attaching of sister chromatids of each chromosome at the centromere. That happens during what phase? Sister chromatids are attached. That is something that happens earlier, no? So that happens in prophase, right? I keep on telling this for the 2028 people also. I finished cell cycle, so I was telling them for them as well. Please study the prophase and prometaphase differences. What happens during those two phases very carefully. Okay, easy. I mean, aparadhi marks if you can't remember. Formation of mitotic spindle begins. Formation of spindle begins in what phase? Formation of mitotic spindle begins. This is something very interesting. Okay, mitotic spindle formation takes place during three phases. It begins during prophase. It occurs during prometaphase. It is completed during metaphase. Okay, begins in prophase. It begins in prophase. What begins in prophase is completed during metaphase when the centrosomes completely reach the opposite poles. Until then, spindle formation is happening. Okay. Centrosomes move towards opposite poles. That I think centrosomes move towards opposite poles. That happens during prophase as well. Spindle microtubules get depolymerized. There is something that happened in telophase, right? Histochromatids of each chromosome at their centromere. Chromosomes move towards opposite poles. Centrosomes start to move towards opposite poles. A small issue there, no? Question number 27. There's a small issue, no? Attaching of sister chromatids of each chromosome at their centromere. No, that is not correct. Neither. We have to give all for this. Yeah. Because nothing happening in metaphase is given, right?
If they said centromeres are aligned at the metaphase plate, then that is okay. Yeah, take all for 27, okay? 27 all. All right. What about fourth answer? Centrosomes move towards opposite poles. Centrosomes move towards opposite poles. That happens in prophase, right? They start to move towards opposite poles. Centrosomes move towards opposite poles due to the lengthening of their microtubules. That happens in prophase. Okay, that happens in prophase, not during meta. Okay, you can take over. Right, 28. Okay, the most accurate regarding mechanism of action of enzyme. Okay, mechanism of enzyme action. 28. Enzyme catalyzes one or few reactions. No, enzymes are reaction specific. Reaction specific. Active site of an enzyme is fully complementary to the shape of its specific substrate. No. Yes. What can you say? Active site is fully complementary. That is the old theory. That is what we call as the lock and key. No? So we don't actually currently accept the lock and key hypothesis of enzyme specificity. We don't currently accept that. What we currently accept is what? What is the theory that we currently accept? Induced fit. That means these are not very rigid things. Whenever the substrate comes towards the active site changes its shape slightly so that they can have a very nice close attachment. Fully complementary, no. Protein components essential for catalytic action of some enzymes are called cofactors. No, they are always non-protein. Non-protein components essential for catalytic action, they are called cofactors. So you can correct it, they are non-protein components. Few amino acids from the active site, yes, few specialized amino acids are found in the active site, right? And those amino acids can interact with the substrate molecule so that the reaction can happen. Reversible cofactors are called coenzymes. No, coenzymes are what? Organic cofactors. Organic cofactors. You can write in front. Organic cofactors are called coenzymes. Moving on, question number 29. 4. Sorry, question number 29, let's see. Okay. Select the correct statement regarding anaerobic respiration. Okay. Some bacteria do ethyl alcohol fermentation. Some bacteria do ethyl alcohol fermentation. What do you think? Yeah, right. Anaerobic bacteria, right, can do it. I mean, okay. Then, so that is some bacteria. Yeah, we have to sort of go with the resource book given things. Okay, අපි එක තියලා right? Limitation of NAD plus increases recycling of NADH and utilization of NAD plus. What do you think?
limitation of NAD, okay, NAD is limited, increases the recycling of NADH and utilization of NAD plus. Not really. So there's a limitation of NAD, that is correct. That doesn't increase the recycling of NADH, but we use NADH in a wasteful way. We use NADH in a wasteful way. What do? You convert pyruvate, you convert acetaldehyde into ethanol using the NADH, right? So that is actually our main goal is not to produce ethanol. Our main goal is to do what? To recycle NADH into NAD plus, because you need NAD plus if you want to keep doing glycolysis, right? So that is not the main reason, right? So for NADH is recycled by using it to convert acetaldehyde into alcohol. Lactic acid fermentation is a process that produces ATP in the presence as well as absence. No, it is, if you are doing some sort of fermentation, that is always anaerobic. In ethyl alcohol fermentation process, the final electron acceptor is acetaldehyde. In final electron acceptor is called, yes, as well as the final hydrogen acceptor, as well as the final electron acceptor is acetaldehyde. So it will accept the electrons from the NADH. Ratio of the volume of oxygen consumed to the volume of carbon dioxide released by a proteinaceous substrate is 0.7. That is a correct, no wait, it's not correct also, right? So what are they talking about here? Respiratory quotient. Respiratory quotient they are talking. So that means VCO2 divided by VO2. Ne? So they are saying, okay, 0.7. What is the VCO, respiratory quotient for protein? Proteinaceous substrate means protein. What is the respiratory quotient for protein? Whenever you are doing respiration to, yes, it is something in the middle. Ne? Carbohydrates has 1, lipids have 0.7, because why? It requires a lot of oxygen to break down lipids. Therefore, the RQ is the lowest for lipids. Protein is something in the middle. So anyway, this is wrong. It is has to be 0.8. At the same time, that's something we are talking about in aerobic. Ne? Then we are here we are talking about anaerobic. So you can't take this. So methanadi, we have to rely on a bit of a technicality. Methana resource book says many bacteria do ethyl alcohol fermentation. But anyway, this is a very good answer. No? Answer number 4 is a no doubt correct answer. No? So we'll take it as the answer. Question number 30. Okay. Natural classification differs from artificial classification due to, what do you think? A system developed before the study of evolution, that is artificial classification, no? Based on convenience, right? Things you can see from outside without considering their natural evolutionary uh, relationships. Easy to expand by adding more groups, that is also artificial. Based on many characteristics, that is natural. Based on a few pre-selected unifying characters, that is artificial. Common characteristics, that is artificial. Okay. In natural classification, you take into consideration so many things, right? That's why it's more difficult. It's not convenient to do natural classification. Okay.
Any questions? So we have reached question number 31. Any questions up until now? Break account, though we'll finish a bit early. Break account, any? We'll finish a bit early. Okay, right. Let's continue. 31. Based on the following statements, okay, bony skeleton being the first group to possess limbs, ectothermic, lungs for respiration. Yeah, I am having a bony skeleton. I am the first to possess limbs. Who am I? I am ectothermic. Who am I? Yes. Bony skeletons. That means you are removing chondrichthyes. Possess limbs. So someone, of course, then fishes are out, right? First ones to have fish, then you can get the answer. It has to be a certain amphibian. In amphibians, of course, they don't have a fixed body temperature, and they usually have lungs, right? Toad, right? Easy peasy. Okay. 32. Correct statement about meristematic tissues is, okay. Can you hear me? Yeah. Any issues? We finished early. Okay. Right. Okay. Correct statement, truth statement regarding meristematic tissues. All new cells formed during cell differentiation undergo elongation and differentiation. 32. All new cells undergo elongation and differentiation. I agree with this differentiation, that is correct. But do they all undergo elongation? Do they all undergo elongation? Can you give me an example of a cell that is arising from meristematic tissue that will not undergo elongation but will undergo differentiation? Yeah. Root cap cells. Root cap, from the root apical meristem, it produces two cells, no? To the outside and to the inside. To the outside cells becomes root cap, right? So root cap cells, they don't elongate. Meristems can have dormant periods, yes, because meristems divide only when the conditions are good. The plant is growing by meristematic tissues. No? So the plant will not grow in unfavorable conditions. So those kind of conditions, the plant stays dormant. Some of them, only some of their cells have a dense cytoplasm. No, they are very active in metabolism because they are dividing fast. Periderm is formed as a result of primary growth. What do you think? No, periderm is a result of secondary growth, right? With the cork layers, okay, and the cork tissue, you get the periderm. Apical meristems contribute to rapid regrowth in damaged leaves. Damaged leaves are repaired by what? Can anyone tell me? Sleepy නැති වෙන්න answer එකක් දෙන්න බලන්න. What meristem repairs damaged leaves? What meristem repairs damaged leaves? Yes. Increase internode length. Ah, yes. Very good. Intercalary. Intercalary meristems. 33. We'll discuss up to 35 and we'll stop for the day. We have to have another session for the SCQs. Okay, we have to have another session and for the remaining MCQs. So that will happen next Friday. Okay, that will happen next Friday and go through the mistakes you might have done in the marked papers after they have been submitted. Okay, so don't worry if you didn't have the paper today for the MCQ discussion because we are going to discuss only 35. For the structure is you are going to have the paper in your hand. Okay, people who didn't receive the paper. Okay, right. Mature cells of parenchyma bear only primary cell walls. Yeah, parenchyma are living cells. So once you get a secondary cell wall, that means you are going to die. So that is correct. Collenchyma are elongated and having flexible cell walls. That is correct. Sclerites are seen in plants where the growth has completely stopped. What do you think? Yes. Sclerenchymas are dead cells, no, so they are never found in sort of actively growing places. So it is C. Sorry, answer number 5. All A, B, C, yeah, correct. Okay. 34. Which of the following processors is important for the opening of stomata during the day? Decrease in CO2 concentration in the substomatal cavity. Yes, when the CO2 concentration reduces, in order to take CO2, stomata opens. Loss of K plus from guard cells. Banning. If you lose K plus, what's going to happen? If you lose K plus out to the outside, water is going to follow. When water is lost, the turgidity is lost. So that the guard cells, they were like this, that were open, they are going to become more flat, so that the stomata closes. That is false. High temperature is going to what? Usually close the stomata. And high temperature is usually going to close the stomata. Okay, so that is also false. A only, right? 34, answer number 1. A only. 35 kalla, we'll stop for the day. Okay, paper discussions continuously can be exhausting. We'll have another session next week and we'll completely cover. Okay, any questions? 33, some mature parenchyma do have. Which parenchyma has a secondary cell wall? Any mature parenchyma? The theory is that the secondary cell wall is an impermeable, very rigid structure. No? Once you get an impermeable structure surrounding you, you can't live because you can't get water, materials. Right? It's very difficult. I mean, you can't survive with a secondary cell wall. Yes. Okay. which is possible to show deficient symptoms such as healthy appearance, slow development, thin stems, purpling of veins. Direct theory point, it's phosphorus. Healthy appearance but slow development, right? It looks okay, but right? So we'll understand when you're doing microbiology, phosphorus is actually the most limiting nutrient for a plant. Very, very difficult to get phosphorus as a plant. It's a difficult life. Okay. You need a way to solubilize phosphate in your soil, that is done by phosphate solubilizing bacteria in the soil. Without those bacteria, the plants can't get their phosphorus. Same for nitrogen. Nitrogen is available in the atmosphere as N2. Very difficult to break down N2, right? You know how stable the nitrogen molecule is, Mehemet? Very difficult to break this triple bond. For that, you require enzymes, nitrogenase, that is found in certain nitrogen fixing bacteria. Okay. Right. So we'll stop there for the day. We have few more MCQs to discuss, and we'll join with the structured essay discussion next week. So next week at the same time, paper class time at 5 p.m. At same paper class time is actually 5:30. We are going to start a bit early because the paper is long. At 5, we'll start paper discussion. Okay. Then any questions if you have, you can ask. How was the paper, right? You will get the marks and you can check. Okay, there's some. Okay, there's a doubt. Let me check. Most of the cells lack secondary cellulose, right? That Right. Most of the cells lack secondary cellulose, right? In within among the parenchyma, okay, among parenchyma, there can be one or more few cells that has secondary cell walls that are dead. Okay, that is because, for example, if you take like a coarse fruit, right? The coarse fruit can contain. I mean, have you seen sort of a certain types of pears are there that are having sort of like a valley vage texture, right? So the fruit of the flesh of the pear is mostly parenchyma, but it can have in the middle can have a bit of sclerites. So they can have secondary cellulose. So the parenchyma is mostly having only primary cellulose. Very few can have secondary cellulose here and there. But remember, if you have a secondary cellulose, you can't live. It's not a living cell anymore. Secondary cellulose are impermeable. Okay. Okay, right. Anything else? Anything else? Okay, then we'll finish for the day. Okay. Join with the paper discussion next week as well. This Sunday, we are going ahead with circulation. So you can join with the people who are going to watch this recording. Okay. I hope you reach this point in the recording. Okay. Then check your mistakes, cover everything, anything you haven't studied properly. Okay. Self evaluation is the most important thing. Right. Okay. Then thank you for joining. Good night.